Welcome back to the deep dive. Today we are talking about a system of such incredible complexity and power that honestly it's almost impossible to truly grasp. I mean, every moment of every day, your body's immune system is executing this perfectly orchestrated war, fighting off viruses, bacteria, potential tumor cells, all with a precision that, well, no machine could ever replicate.
It really is a marvel of evolution,
Right.
absolutely. But if you stop and think about that immense, constantly running power, this vast army of immune cells designed specifically to identify and destroy anything foreign, the central paradox becomes pretty glaring.
Right.
If the system is so potent, how does it manage, most of the time, to avoid turning that power inward? How does it stop itself from destroying the very tissues and organs it's supposed to protect?
Yeah, that question, the mystery of immune self-tolerance, that's really what defines the fundamental biological breakthrough we're covering today. We are unpacking the 2025 Nobel Prize in Physiology or Medicine, awarded to Mary E. Brunkow, Fred Ramsdell, and Shimon Sakaguchi for identifying the body's ultimate, let's call them security guards.
hmm Their combined work provided the essential knowledge that solved the puzzle of peripheral immune tolerance. And we're talking about the second absolutely critical layer of defense here. This is the one that guarantees that even when the immune system is fully activated, fully armed, it tolerates the body's own tissues. And this wasn't just, fixing a minor scientific oversight. It fundamentally defined a whole new field. It paved the way for targeted treatments in cancer, autoimmunity, transplantation, treatments that were just impossible before.
So before we can really appreciate the scale of their breakthrough, we need to set the scene a bit. You have to understand the immense difficulty, the sheer sort of mathematical challenge the immune system faces, and, well, what the scientific community thought was the solution way back in the 1980s.
Yeah, the context is key.
All right, so our story of self-tolerance, it really revolves entirely around one type of cell, the T cell, or lymphocyte. These are the specialized sentries, you could say. They're born in the bone marrow, they mature in the thymus, and they're absolutely central to adaptive immunity.
Exactly, and T cells come in different specialized flavors, each with a critical role. For our story today, we primarily care about two types, the helper T cells and the killer T cells. Helper T cells are kind of like the generals. They coordinate the whole attack. They recognize threats. They signal other cells to mobilize.
Right.
Killer T cells, they're the sharpshooters. Their job is to identify and basically eradicate cells that have been infected by viruses or pathogens or cells that have gone rogue, like tumor cells.
And how do they do this incredibly sophisticated surveillance? They use these specialized proteins on their surface called T cell receptors. Think of them like millions of tiny specific antennae constantly sampling little bits of protein presented on the surface of every cell they bump into.
And the way the body creates these receptors is, well, it's the marvel, but it's also the central problem we were talking about. T cell receptors aren't pre-programmed with specific targets. They're created through this process called VDJ recombination. You can almost picture it like a biological slot machine, where various gene segments, variable diversity, and joining segments are randomly shuffled and combined as each T cell develops.
And the sheer number of possible combinations is just staggering.
It really is. The theoretical number is mind-bogglingly vast. It could exceed 10 to the power of 15. That's a 1 with 15 zeros after it. 10, 15 zeros after it. 10, 15 unique receptors. This huge library is basically the body's guarantee that no matter what threat pops up, a new flu strain, some exotic pathogen we've never seen, even a completely novel virus like the one that caused COVID-19 back in 2019, there's always likely to be a T cell somewhere that can recognize it and kick off a response. This randomness is the secret sauce for immune flexibility.
But that flexibility comes at a cost, doesn't it? That massive random generation process. It means the body must also, just by pure chance, generate T cells whose receptors perfectly match and recognize the proteins found naturally on our own healthy tissues, our endogenous proteins.
Exactly. It's an unavoidable side effect of having such a comprehensive defense
That's
system.
the inevitable flaw, as you said.
It is. If you generate 10, 15 potential keys, it's almost certain some of them are going to fit your own locks. And this is why the accepted mechanism for preventing immediate self-destruction, going way back to the 1980s, was something called central tolerance.
Right. Describe that old theory for us. The idea was basically that these potential rogue T cells, the ones that could attack our own body, were caught early, before they ever got out into the bloodstream.
Precisely. The T cells mature in the thymus, that's a gland located in your chest, behind the breastbone. While they're developing there, they undergo this really rigorous screening process, like a boot camp. First, they get checked to make sure their receptors actually work, that they can bind to things properly. That's called positive selection. Then comes the crucial part, negative selection. Any developing T cell that shows strong recognition, a strong binding affinity for the body's own proteins, the self proteins presented to them in the thymus, is eliminated.
Oh.
They're triggered to undergo apoptosis, which is basically programmed cell death. They commit cellular suicide.
So the military boot camp theory, weed out the potential traitors, the self-reactive cell, while they're still in basic training in the thymus. That's it. Simple.
That was the widely accepted dogma, yes.
Yeah.
And historically, this view was actually heavily reinforced by some earlier scientific dead ends. Back in the 1970s, some researchers had sort of played with the idea of a second layer of control, maybe a dedicated cell type that actively suppressed the immune response. They called them suppressor T cells.
Ah, I've heard of those. But the evidence for those initial suppressor T cells was messy, wasn't it?
It was very messy, often inconsistent, difficult to reproduce, and sometimes based on experimental methods that turned out to be flawed. So when the concept of central tolerance came along, offering this neat, simple, elegant answer, just eliminate the rogue cells, the more complex, frankly confusing idea of active suppression got completely thrown out. Discredited, really.
Yeah.
The consensus became, if a T cell attacks self-tissue out in the body, it must represent a failure of central tolerance in the thymus, period. There was no backup system thought to exist.
So the prevailing mood in the scientific community, say, in the early 1990s, was that immune self-tolerance was this all-or-nothing process, executed entirely in the thymus, done and dusted. But this clean, elegant theory missed one fundamental kind of stubborn piece of evidence, something that suggested the story wasn't quite over.
Exactly. And that stubborn piece of evidence really captivated our first laureate, Shimon Sakaguchi, who was working in Japan at the Aichi Cancer Center in Nagoya. Unlike most of his peers at the time, Sakaguchi just couldn't let go of the idea that there had to be some kind of active regulation happening out in the body, a mechanism beyond just deleting cells in the thymus. He was really swimming against a very strong scientific current back then.
And his main clue, his primary evidence, came from this kind of paradoxical mouse experiment performed by his own colleagues, right? This is a great example of science progressing not from proving a hypothesis, but from trying to explain a result that just didn't fit.
Precisely. Sakaguchi's colleagues were studying the thymus and its role in immunity. They were performing surgical removals, thymectomies, on newborn mice.
Yeah.
Their straightforward hypothesis was, okay, if we remove the T cell factory, the thymus, these mice should have fewer, weaker T cells and therefore a compromised immune system. Pretty logical.
Makes sense.
But what they observed was this really weird timing anomaly.
Okay, tell us about the timing difference.
Well, if they removed the thymus right after birth, say day 1, the mice often survived, although yes, they were immunocompromised as expected. But if they waited just a couple of days and did the procedure 3 days after birth, the results were absolutely catastrophic. These day 3 thymectomized mice developed these raging systemic autoimmune diseases. Severe inflammation of the gut, their thyroid glands were attacked, their spleen and lymph nodes became massively enlarged and sickly. Their immune system wasn't weak, it was dangerously hyper-aggressive against their own bodies.
Wow. So those first 3 days were absolutely critical. Whatever mechanism normally protected the body from this kind of self-attack had apparently already been established and deployed into the circulation within those first 72 hours. Removing the thymus after that point meant you lost the factory, sure, but the cells that were already out there, they just went completely rogue without whatever was regulating them.
That was exactly Sakaguchi's immediate conclusion. It couldn't just be about deleting T cells back in the thymus. There had to be a specific population of cells released from the thymus during those crucial first 3 days that actively regulated, actively controlled the rest of the immune system out in the periphery. These were the missing security guards.
So he set out to prove their existence with his isolation experiment. This sounds crucial.
Yes, it was definitive. To prove this active regulation existed, Sakaguchi performed a really elegant experiment. He took T cells from genetically identical, healthy adult mice, normal mice. He then carefully separated this mixed population of T cells, and he injected them into those mice that had their thymus removed 3 days after birth, the mice that were otherwise guaranteed to develop that raging autoimmunity.
Okay, so he's transferring T cells from a healthy mouse into one that's destined for autoimmune disaster. What happened?
It protected them. The injection prevented the disease. The mice that should have succumbed to severe self-attacking illness were largely protected by the total population of T cells he transferred. And critically, he did control experiments. If he transferred only the potentially attacking T cells, which he could isolate based on certain markers, The mice still got sick. But when he transferred the full mixed population from the healthy mouse, the disease was blocked.
Oh, so that proved it.
It proved, fundamentally, that within that mixed population, there was one type of cell actively restraining, actively suppressing the other potentially dangerous cells.
That is a phenomenal conceptual leap right there. It showed that immune regulation wasn't just passive elimination back at headquarters. It was active control out on the streets, so to speak.
It absolutely did. But now came the really hard part, a challenge that took him about a decade. Identifying the specific molecular signature of this restraining cell. How do you pick it out of the crowd? Researchers already knew that these cells were T cells, and they carried the surface marker CD4, which was confusing because CD4 is the marker usually found on helper T cells, the ones that are supposed to activate the immune system.
Right, so you had cells that looked like the commanders, CD4 positive, but they were actually acting like peacekeepers or suppressors. They were kind of the wolves in sheep's clothing, biologically speaking.
Exactly. That was the paradox. So Sakaguchi spent the next 10 years meticulously trying to find the unique molecular tag, some other protein on the surface, that differentiated these regulatory CD4 cells from the much larger population of conventional CD4 helper T cells. It was incredibly painstaking work, separating cell populations using various antibodies and assays, and then testing their function. Did they suppress or did they activate?
And he finally nailed it in 1995, found the key differentiator.
Yeah. Yes. In a landmark paper published that year, he showed that these calming suppressive T cells carried not only CD4, but also consistently expressed high levels of a second protein marker, CD25. This subtle yet consistent difference being CD4 positive and CD25 positive allowed him to finally isolate, purify, and characterize this new class of cell. He formally named them regulatory T cells, or TREGs for short.
Okay, so by 1995, we have the physiological proof from the mouse experiments, and we have the cellular marker, CD25, for Sakaguchi's security guard cell. But you mentioned skepticism remained high, partly because the field had been burned before with those old suppressor T-cell ideas.
Yeah.
But what were the critics still demanding?
They needed the why. Okay, these CD4 plus CD25 plus cells suppress things, but how? What was the underlying molecular mechanism? What made them act differently? Crucially, what was their master control switch inside the cell? That was the missing piece.
And the answer to that molecular question, the why, would come from a completely different line of research, starting with a historical quirk involving mice in Oak Ridge, Tennessee, a place connected way back to the Manhattan Project, oddly enough.
That's right. This part of the story begins with a specific, naturally occurring mutant mouse strain known as the scurfy mouse. For years, labs had noticed that male mice born with this particular genetic trait were intensely sickly. They exhibited chronic, widespread inflammation. They had scaly skin, which is where the name scurfy came from. And very characteristically, they had massively enlarged organs. Their spleen and lymph nodes were just engorged with runaway immune cells. They typically didn't live long, only a few weeks.
And this fatal illness was clearly a classic case of the immune system rebelling against the mouse's own body, a total breakdown of self-tolerance.
Absolutely. It was a violent, systemic autoimmune attack. And crucially, genetic studies had already shown that this mutation was X-linked. That means the faulty gene was located on the X chromosome. This explained why it primarily affected male mice, who only have one X chromosome. If they inherit that faulty one, they get the disease. Females with two X chromosomes usually have a backup healthy copy.
And that X linkage was a huge clue, wasn't it? It strongly suggested that maybe a single gene, when mutated, was responsible for this catastrophic failure of immune regulation.
Exactly. It pointed towards a potential master switch.
Okay, now let's bring in our other laureates, Mary Brunkow and Fred Ramsdell. They were working together at a biotech company called Celtech Chiroscience up in Bothell, Washington. Their focus was on developing new treatments for autoimmune diseases, and they instantly recognized the unique power of this scurfy mouse model.
They saw it immediately. They realized that the key to understanding potentially all autoimmune diseases might be hidden within this single faulty gene in the scurfy mouse. If they could just find that one mutation, identify that gene, they could potentially uncover the master molecular switch controlling peripheral immune tolerance in general. It was a huge gamble, but the potential payoff was immense. So they decided to dedicate themselves to finding the specific gene responsible for the scurfy condition.
We really have to emphasize the timeline here again. This was the mid to late 1990s. This was not the era of cheap, rapid, whole genome sequencing like we have today, finding one specific mutation back then.
Oh, it was a monumental task, like finding a single typo in a massive unindexed encyclopedia set. The mouse X chromosome contains about 170 million base pairs, 170 million letters of genetic code. The computational power, the sequencing technology, it just wasn't there yet for a quick search. Brinkow and Ramsdell first had to use these complex, incredibly laborious genetic linkage analysis techniques, basically using known genetic landmarks to narrow down the region on the X chromosome where the mutation had to be.
How far did they manage to narrow down that search area? From 170 million bases to what?
They managed, after a lot of work, to narrow it down to a region containing approximately 500,000 nucleotides. 500,000 bases. Still massive by today's standards, but, a manageable target for the time.
Still sounds pretty daunting.
It was. Their work then became this really grueling exercise in detailed molecular mapping. They had to physically order and map out the segments of DNA within that 500,000 base pair region, and then for every potential gene they identified within that segment, they had to sequence it meticulously in both healthy mice and the sick scurfy mice, looking for that one critical difference, that one mutation.
It sounds like a true scientific marathon, just fueled by sheer determination and probably a lot of late nights in the lab. They must have been comparing dozens of potential candidate genes within that region.
They identified 20 potential genes within that narrowed down segment, 20 needles in a haystack, even if it was a smaller haystack now. Each one required detailed, dedicated sequencing and comparison work. You can just imagine the tension as they systematically went through the list, checking gene number 1, comparing sequences, nothing. Gene 2, normal. Gene 3, fine, all the way through.
But the faulty gene, it turned out to be the very last one they checked, didn't it? Talk about persistence paying off.
Unbelievably, yes. It was only the 20th and final candidate gene on their list that finally gave them their definitive breakthrough. Bingo. They found a specific single base pair mutation, a tiny change in the genetic code, in a gene that, at the time, was previously unknown. They did notice, however, that this new gene had structural similarities to a known family of genes called the FOX genes, which stands for fork-head box. These FOX genes were known to act as transcription factors.
Transcription factors, those are the genes that control the activity of other genes, right, like master switches.
Exactly. They bind to DNA and regulate whether other genes get turned on or off. So finding a mutation in a FOX-like gene made perfect sense in the context of such profound immune system failure. They named their newly discovered gene FOXP3.
Wow. A completely unknown transcription factor revealed as the direct cause of catastrophic immune failure in these mice. That's a fundamental discovery in itself. But their work didn't stop there. They immediately suspected and then confirmed the human connection.
Yes, the symptoms of the scurfy nose, the severe autoimmunity, the X-linked inheritance were almost a perfect mirror image of a rare but devastating human autoimmune condition called IPEX syndrome. IPEX stands for immune dysregulation, polyendocrinopathy, and neuropathy, X-linked syndrome. It's a mouthful, but it describes boys born with similarly catastrophic multi-organ immune attacks. They often suffered from severe diabetes, thyroid problems, gut inflammation, and tragically often die in infancy or early childhood without treatment like bone marrow transplant.
So Brunkow and Ramsdell must have raced to check if the human version of this new FOXP3 gene was also mutated in boys suffering from IPEX.
They did exactly that. Working rapidly with pediatric immunologists and clinicians, they managed to collect DNA samples from several unrelated boys who had been diagnosed with IPEX syndrome. They sequenced the human equivalent of the gene, which they called FOXP3, all caps for the human gene, in these patients, and they confirmed it. Harmful functional loss mutations in the human FOXP3 gene were indeed the direct cause of IPEX syndrome.
Incredible. So their findings, published back-to-back in Nature Genetics in 2001, provided the critical molecular mechanism. They proved that this single specific gene, FOXP3, was the absolute linchpin responsible for preventing the severe, fatal breakdown in immune self-tolerance in both mice and humans. We now had Sakaguchi's regulatory cell, the TREG, and Brunkow and Ramsdell's master gene, FOXP3. The pieces were falling into place.
And the final crucial synthesis of all this work happened very quickly after that, around 2003. Several research groups, importantly including Sakaguchi's own lab, conclusively demonstrated the definitive link. They prove that the FOXP3 gene, the molecular switch discovered by Bronkow and Ramsdell, is the absolute master regulator that dictates the development, the function, and even the stability of those regulatory T cells that Sakaguchi had identified 8 years earlier.
Okay, so let's break that down. If a developing T cell, say in the thymus or perhaps even later, starts expressing high levels of the FOXP3 protein inside its nucleus,
It gets programmed to become a regulatory T cell. Its fate is sealed. If it doesn't express significant FOXP3, it proceeds down other pathways, maybe becoming a helper T cell or a killer T cell.
so FOXP3 acts like the foreman on a construction site, telling that cell what job to do.
That's a good analogy. FOXP3, being a transcription factor, sits in the cell nucleus and binds to DNA. It essentially turns on a whole specific genetic program, activating dozens of other genes while shutting down others. By initiating this unique program, FOXP3 fundamentally shifts the cell's identity. It changes it from a potential attacker into a dedicated peacekeeper. It programs the cell to become a master suppressor of immune responses.
And establishing this direct link, FOXP3 controls TREGs. This is what fundamentally defined peripheral immune tolerance, didn't it? It showed it wasn't just some vague phenomenon, but a clearly defined active biological process, driven by a specific gene executed by a specific cell type.
Exactly. It showed that central tolerance, the deletion in the thymus, while definitely important, was only the first line of defense. It wasn't the whole story.
Peripheral tolerance, mediated by FOXP3 and TREGs, is the essential backup system, the ultimate insurance policy, really.
It absolutely is. And this system, driven by TREGs, has a vital dual function. The first function is the one we've mostly been talking about. These TREGs act as the security force constantly patrolling the circulation in tissues. Their job is to ensure that any potentially self-reactive T cells, those that inevitably slip past the screening process in the thymus, are actively restrained and prevented from launching an attack against the body's own tissues.
The existence of those slippage T cells is really important, isn't it? It implies that the central tolerance system in the thymus is inherently leaky, or maybe can't possibly screen against every self-protein perfectly.
That seems to be the case. The sheer diversity needed in the T cell receptor repertoire to fight off any potential external threat makes it practically impossible to achieve perfect elimination of all self-reactivity during development. There's always going to be some low-level potential for autoimmunity. Therefore, the body absolutely relies on these active security guards, the FOXP3-driven TREGs out in the periphery, to manage that constant low-level threat and maintain peace.
Okay, that's function one, preventing accidental self-attack.
Huh.
What's the second key role of these regulatory T cells?
The second function is perhaps just as crucial, especially for long-term health after you've fought off an infection. It's the shutdown mechanism. Think about it. When you have a major infection, say a bad flu virus, your immune system ramps up dramatically. T cells multiply rapidly, they become highly activated, they operate at peak intensity. This causes significant inflammation. That's the fever, the aches, the swelling you feel. It's necessary collateral damage to fight the invader.
Right.
But once the pathogen is successfully eliminated, the body needs an immediate and effective way to stand down that hyper-aggressive immune response. You can't just leave the system running at full throttle indefinitely. Regulatory T cells play a critical role here. They step in to calm things down, ensuring the immune system doesn't continue working at top speed, which would otherwise lead to chronic inflammatory damage or even trigger autoimmunity in the aftermath of the infection.
That detail really changes how you think about the immune system's design, doesn't it? It's not just about detection and destruction. It's equally about control, moderation, and resolution. And Laureate's work proved that this control isn't just a passive winding down. It's an active, genetically coded process driven by FOXP3 and TREGs. This discovery just immediately transformed immunology. It shifted the focus from simply maximizing the attack to figuring out how to achieve the perfect balance.
And that transformation, that new understanding of balance, immediately opened up countless avenues for medical applications. It was like finding the immune system's dimmer switch. For the first time, researchers had a specific molecular lever, this FOXP3 TREG axis, that they could potentially manipulate to tune the immune system up or down as needed. And we are now seeing treatments based on this knowledge advancing rapidly through clinical trials, generally following two opposing strategies depending on the disease you're trying to treat.
Okay, let's start with cancer. Here, the goal is usually the opposite of tolerance you want the immune system to attack. So the strategy involves decreasing the action of these regulatory T cells, right? Undermining the security guards.
Exactly. Tumors, being the devious biological entities they are, have actually figured out how to exploit the body's own self-regulatory mechanisms to protect themselves. A lot of research has now definitively shown that many solid tumors, think melanoma, breast cancer, colon cancer, many others, are often heavily infiltrated by or surrounded by a high concentration of regulatory T cells. These TREGs are actively recruited to the tumor environment by specific chemical signals the tumor releases.
So the tumor essentially hijacks the body's own peacekeeping force, co-ops the security system to build a protective shield around itself.
That's precisely what happens. Those regulatory T cells, operating under the command of FOXP3 within the tumor microenvironment, actively suppress the local immune response. They release suppressive signaling molecules, cytokines like interleukin-10 and TGF-beta, which effectively paralyze the killer T cells that are trying to infiltrate the tumor and destroy the cancer cells. This creates what immunologists call an immune cold or immunosuppressive microenvironment, basically shielding the cancer from the immune system's attack.
So the therapeutic strategy here is to find ways to specifically neutralize or remove or maybe inhibit the function of those particular TREGs within the tumor area, ideally without crippling the body's overall immune tolerance elsewhere, which would cause autoimmunity. That sounds tricky.
That is absolutely the challenge. Current research is exploring several angles. One approach involves targeted depletion using antibodies, for example, that specifically bind to markers on TREGs, maybe coupled with toxins to try and eliminate them only near the tumor. Another, perhaps more sophisticated strategy involves finding drugs that specifically block the function of TREGs, maybe interfering with FOXP3 activity itself or the suppressive molecules they release. And often the idea is to integrate TREG inhibition into existing immunotherapy regimens. For instance, combining immune checkpoint blockade therapies, which aim to release the brakes on killer T cells, with drugs that simultaneously target TREG signaling pathways. The goal is a coordinated punch. Unleash the attackers while also dismantling the tumor's protective TREG shield.
Okay, that makes sense for cancer. Turn down the suppressors. Now let's pivot to the completely opposite strategy. Treating autoimmune diseases like type 1 diabetes, multiple sclerosis, rheumatoid arthritis, lupus. Here, the problem is too much attack against self, so you want to boost the security guards, the TREGs, to restore tolerance.
Exactly. In these autoimmune conditions, the body's killer T cells, and sometimes helper T cells too, are mistakenly targeting healthy tissues, pancreatic beta cells in diabetes, the myelin sheath around nerves in MS, joint tissues in arthritis. The regulatory T cell system is either insufficient or malfunctioning. So the therapeutic concept is straightforward. Stop the friendly fire by reinforcing the peacekeeping force. Find ways to increase the number or enhance the function of the patient's own regulatory T cells.
And how are researchers trying to achieve that boost in TREGs?
One of the most promising avenues currently in clinical trials involves using a naturally occurring signaling protein called interleukin-2, or IL-2. Now, IL-2 has been known for a long time, and at high doses, it actually stimulates the proliferation of all T cells, including the attacking ones, which can be dangerous. But what scientists discovered, somewhat serendipitously, is that TREGs express a high-affinity receptor for IL-2. This means that very low, carefully controlled doses of IL-2 preferentially stimulate and expand the population of regulatory T cells much more effectively than they boost the effector T cells.
So low-dose IL-2 acts almost like a specific fertilizer for TREGs.
Yeah.
It helps them thrive and multiply, effectively increasing the size and strength of a security force without overstimulating the attackers.
Precisely. It tips the balance back towards suppression and tolerance. Pilot studies using low-dose IL-2 therapy are ongoing for various autoimmune conditions, and they've shown that it can safely increase TREG populations in patients and, in some cases, lead to clinical improvement. It offers a potentially elegant way to restore the broken immune balance naturally, rather than just hitting the whole system with broad immunosuppressants.
And this leads us directly into the third major area of application, transplantation. The challenge here is very similar to autoimmunity. You need to prevent the immune system from attacking something it sees as foreign, in this case, the transplanted organ.
Right. Transplant rejection is fundamentally a powerful immune attack against the donor organ. The recipient's T cells recognize the proteins on the transplanted liver or kidney or heart as non-self and mobilize to destroy it. Traditionally, this has been managed with powerful, broad immunosuppressive drugs, things like cyclosporine or tacrolimus. These drugs work, but they essentially mute the entire immune system. They hit it with a sledgehammer. This leaves the transplant recipient highly vulnerable to opportunistic infections and even increases their long-term risk of developing certain cancers because the immune surveillance function is dampened everywhere.
But the knowledge gained from Brunkow, Ramsdell, and Sakaguchi suggests a much more targeted pathway, right? A way towards achieving specific tolerance just for the new organ, moving away from that dangerous blanket immunosuppression.
Absolutely. This is where the field gets really exciting, entering the realm of advanced cellular engineering, building directly on the TREG discovery. Researchers are now actively practicing sophisticated forms of cellular therapy. The basic idea is you isolate a patient's own regulatory T cells from their blood. You then expand them exponentially in the laboratory, grow them into the billions, creating this massive army of supercharged peacekeepers. And then you infuse these expanded autologous, meaning the patient's own, TREGs back into the patient, often around the time of the transplant surgery. The hope is that this flood of TREGs will induce tolerance specifically to the new organ.
And the most cutting-edge research takes that concept even one giant step further, doesn't it? Integrating insights from cancer immunotherapy like CAR-RT cells, but flipping the script. We're talking about creating engineered TREGs, sometimes called CAR-TREGs.
Yes, exactly. It's a brilliant adaptation. You take the concept used in CAR-RT cell therapy for cancer, where you engineer killer T cells with a chimeric antigen receptor, or DARR, to make them specifically target a protein on tumor cells. But here, you engineer regulatory T cells with a CAR designed to recognize a specific target on the transplanted organ.
So instead of creating a CAR-T cell that's a guided missile to kill cancer, you're creating a CAR-TREG that's like a guided shield, specifically programmed to protect the new organ.
That's the idea. The CAR acts as a highly specific address label or homing device. It's engineered to recognize a protein that is present on the donor organ, but not widely elsewhere in the recipient's body, maybe an allantogen, one of those proteins that differs between donor and recipient. When these engineered CAR-TREGs are infused back into the patient, they should theoretically travel directly to the transplanted kidney or liver, bind to it via their CRR, and concentrate their suppressive activity right there, at the site of potential attack.
That is just an astonishing level of potential precision. You're sending a dedicated security detail right to the specific organ that needs protection, instructing them to keep the peace locally while leaving the rest of the immune system fully functional to fight off actual threats like viruses and bacteria. This feels like moving from the equivalent of chemotherapy for the immune system broad suppression to something more like laser-targeted radiation therapy, hitting only the necessary spot.
It really is the potential fulfillment of the promise inherent in discovering peripheral tolerance. Sakaguchi showed the crucial cell existed. Brunko and Ramsdell found its master switch, FOXP3. And now medical science is figuring out how to build the control panel, how to manipulate that switch and direct those cells with unprecedented accuracy. This discovery didn't just reveal a fascinating piece of biology, it gave us the fundamental blueprint for potentially reprogramming the human immune response for therapeutic benefit. #tag outro.
What a truly profound journey we've taken today. From a puzzling observation in newborn mice all those years ago to now designing custom-made cellular security forces with specific delivery addresses. This Nobel Prize really rightfully recognizes three distinct yet perfectly complementary contributions that together launched an entirely new era in medicine and immunology.
Absolutely. We honor Shimon Sakaguchi for that crucial initial physiological insight, for persevering against the grain, and for the meticulous work isolating the specific regulatory T cell marked by CD4 and CD25, proving way back in 1995 that active immune suppression was a biological reality.
And then we recognize Mary Brunko and Fred Ramsdell for that incredibly grueling, decade-defining molecular detective work, solving the mystery of the scurfy mouse and, through that, identifying the absolutely crucial master control gene for tolerance, FOXP3, publishing that link in 2001.
And it was the convergence of their work, really cemented around 2003, that established FOXP3 as the definitive molecular blueprint for regulatory T cells and thus for peripheral immune tolerance. It confirmed the existence and the mechanism of this essential backup security system, the one vital for our survival. Their fundamental knowledge truly has, and will continue to, confer the greatest benefit to humankind, as the Nobel Committee states, by providing the essential map for these targeted immune therapies that are now actively fighting cancer, autoimmunity, and completely revolutionizing the field of transplantation.
And as we wrap up, reflecting on the stunning precision of these emerging cellular therapies, especially those CR-TREGs engineered with specific address labels, it really sets up an incredible question for you, the listener, to consider. If we are now gaining the ability to customize the body's natural security guards, to send them with almost surgical precision only where they are needed, maybe to protect a new organ, or perhaps to contain a specific autoimmune attack in one tissue, what does that mean for the future of those old-school, broad, systemic immunosuppressive drugs?
It feels like we are genuinely entering an era where biological engineering, using the body's own finely tuned mechanisms, is poised to replace cruder chemical suppression. The fundamental challenge and the incredible opportunity for the next generation of researchers is built entirely on the foundation laid by Brenkow, Ramsdell, and Sakaguchi. Can we achieve immune tolerance that is so complete, so specific, and so precisely targeted that the significant collateral damage caused by blanket immune suppression becomes just a painful memory of medicine's past? That's the ultimate goal.